Now let's study about the stem cells. Again, we start with the short movies and then move into the details.
Bone marrow contains many different types of cells, several of which have specialized roles in the blood. In the early 1960s, it was shown that if bone marrow was taken from one mouse and transplanted into another, rare and specialized cells could travel to the spleen where they would divide and form small colonies. These colonies contained a mixture of different blood cell types. Incredibly, when these colonies were removed and retransplanted, these rare cells formed new colonies in the spleen of a second mouse. These experiments revealed two defining properties of a stem cell. First, a stem cell must be able to self-renew. That is, it must be able to divide and give rise to more cells like itself. Second, it must be able to differentiate. In other words, it must be able to divide and give rise to specialized cells. Since these early experiments, many different types of stem cells have been discovered, and this general definition applies to all of them.
Following fertilization, the developing embryo contains a group of cells known as the inner cell mass that will eventually go on to form all tissues of the body. However, at this early stage of development, these cells have not yet committed to become a specific type of cell and are said to be in an undifferentiated state. If the cells of the inner cell mass are isolated, they can grow indefinitely in a plastic dish and maintain this undifferentiated state. These cells are known as embryonic stem cells, or ES cells. Like cells in the developing embryo, ES cells retain the ability to form any type of cell in the adult body. Thus, ES cells are said to be pluripotent. By studying the signals required to control how ES cells develop into specific cell types, scientists may be able to develop therapies capable of restoring damaged tissues.
Did you get a good idea about the stem cells? Fertilized egg cells develop into embryos. An early stage of blastocyst stage, the inner cell mass can be collected and cultured, and these cells can become embryonic stem cell. Embryonic stem cell can make any kind of tissue cells in the body. That's why the property of this embryonic stem cell we call pluripotent. If one cell type can make only one tissue or one type of cell, we call that unipotent. Now, earlier than blastocyst, this fertilized egg cell stage, to some extent, we have the stage of a totipotent stage. Totipotent is slightly different from pluripotent. Pluripotent cells can make every tissues, but it cannot make individual. But totipotent can make real individual. We'll see those differences later. Here is a little more details. So starting from one fertilized egg cell, it forms blastocyst, and then from the inner cell mass we can make stem cells. So this stem cell can make all the cells. This is a pluripotent embryonic stem cell. And then first step, it differentiates into different germ layers. They are endoderms, mesoderms, and ectoderm. And then they can make other type of intermediate stage of adult stem cells. These stem cells are not pluripotent, but at least they can make multiple numbers of different cell types. That's why a new term we need to describe these adult stem cells. Those adult stem cells are multipotent, we say. And after it is differentiated into the final stages, each cell type can make only one type of cell, then they become unipotent. Our life starts when sperm cell meets egg cells, and then maybe only one sperm cell can enter the egg cells and make a zygote. So maybe here, in this case, this sperm is getting into the egg cells. Maybe this is the winner. This image is a snapshot made by electron microscope. Once fertilized egg cells develop into blastocyst stages, here is inner cell mass. But another important cells around here, these cells are trophoblast. Inner cell mass can make pluripotent cells, but without trophoblast, it cannot make the whole body, whole individual, because this trophoblast make important organs, this placenta. Placenta is a part of embryo tissues and which connect the embryo to the mother side. It's interesting, isn't it? This is the embryo, and embryonic stem cell can make any cells of an embryo, but it does not make a placenta. The trophoblast cells, they make placenta, and they do not give that job to the mother cells. They make their own placenta, attach itself to the mother, and attract the nutrient for himself. So to summarize, inner cell mass can make a pluripotent stem cell, but it becomes totipotent only when it has trophoblast together. This is important. Now let's think about the property of embryonic stem cells. Let's start with the mouse stem cells. All the embryonic stem cells, they can form teratoma. Teratoma is one kind of benign tumors. And embryonic stem cells, they can form all different kinds of embryonic 3-germ layers. Those were endoderm, mesoderm, and ectoderm, and eventually they can make all different types of cells. If embryonic stem cell enters another embryo, they can mix together and carry on their own genetic information and forms some mixtures of life form. This kind of life form we call chimera. So embryonic stem cell can generate chimera. And good quality of embryonic stem cell, once they are entered into different embryo and they transfer its genetic information to the next generation, and the baby should have embryonic stem cells genetic information, then this case we say this is a germline transmissions. This can happen only in good quality of embryonic stem cells. The most difficult test for good quality of embryonic stem cell is tetraploid complementation test. I will explain this in the next page. Now let's think about the human embryonic stem cell. Human embryonic stem cell may have similar properties of mouse embryonic stem cell, but we cannot test chimera formation or germline transmissions because this has serious ethical problems. So once human embryonic stem cell is established, usual test is formation of a teratoma, but not in human, in different animal model like in mouse. Or checking whether the embryonic stem cell can form all 3 different kinds of germ layers. This is a usual test to check whether the cells are really pluripotent or not. Now let's think about tetraploid complementation. If you have sperm cells and egg cells meet together, then they form zygote, and then eventually they make blastocyst and then whole embryos. Now again, this is the inner cell mass and this is the trophoblast. Trophoblast make this placenta, and inner cell mass they make this embryo. In early stage of embryo development, if we give electric shock, now the cells cannot divide properly. Originally, all the cells should have 2 copies of DNA, 2 end stages, but in this case, they becomes 4 end stage. If the embryo becomes a 4 end stage, inner cell mass cannot develop properly and they die out. Only the trophoblast they can survive. In this stage, if we inject embryonic stem cell from outside, then trophoblast provide placenta part, and this pluripotent embryonic stem cell they can provide all the cells for embryo. Trophoblast cells has a tetraploid stage of DNA structure, and it cannot provide embryo. And if embryonic stem cell can make this whole body of an embryo, then this proves that this embryonic stem cell is a perfect pluripotent stem cells. This assay is called as tetraploid complementation assays. Hematopoietic stem cell can make all different type of blood cells. So that's why if we have problem in blood system or by chemotherapy or radiotherapy, if you remove all existing blood system, we need to get new hematopoietic stem cells to rebuild blood systems. Usually the source of these hematopoietic stem cells are obtained from bone marrows, and this technique has been used over half a century. But as you see here, we need to put very thick needles into inside of the bones. It's very painful and also potentially it can be quite dangerous. In this approach, we can inject one kind of growth factor, so-called granulocyte colony stimulating factor. Then the donor produces more hematopoietic stem cells and they release those hematopoietic stem cells into the blood. Then, utilizing their blood and then taking out those blood and by this simple centrifugation method, only collect this embryonic stem cell and recycle this blood back into the donor. So this method is much safer and more convenient than taking their bone marrows. More recently, mesenchymal stem cell has been very popularly being used. Mesenchymal stem cell can make many different types of cells in the body. That's why it has more applications than hematopoietic stem cells. Mesenchymal stem cell also can be obtained from bone marrow, but interestingly, it can be obtained from different sources. Can you guess what source is it? Mesenchymal stem cell can be obtained from extra tissues, maybe the only tissue we want to give out. Those embryonic stem cells can be obtained from our fat tissues. In ancient time, maybe human being, we didn't have enough energies, so we wanted to save a lot of energies and reserve cells, maybe in the fat tissue. And that's why fat tissue has many different kinds of cell types. Mesenchymal stem cell is one of them. So by surgery or liposuction, if you collect this other fat tissue, then among them we can collect a mesenchymal stem cell, and this can be utilized for many different purposes. While hematopoietic stem cell and mesenchymal stem cell have been used most successfully so far, other types of adult stem cells are relatively difficult to collect and also to culture. So that's why, if possible, utilizing the pluripotent property of embryonic stem cells by taking them and then differentiate into different cell types and then utilizing those cells may be the best way. One of the critical problems of embryonic stem cell is they form teratoma. Teratoma is a benign tumor, but you don't want to have unwanted cells in different parts of the body. Another problem can be, once you prepare this embryonic stem cell from other individual, then transfer this into my body, potentially there can be some immune response because our body may recognize other body's cell as non-self. To avoid this kind of immune response, maybe the best way is to make my own stem cells. But embryonic stem cells, they disappear very early stage of embryogenesis. We may lose the chance to recollect those embryonic stem cells. Making the exact same copy of myself, you may call cloning. If you think about normal embryogenesis, we start with one fertilized egg cells. It has a 2N stage of DNA structure, and then it forms blastocyst, and then eventually it develops into embryos and full bodies. Now, this information, the DNA information can decide the property of this individual. Then, maybe one cloning method can be replace this nucleus and inject another nucleus, maybe my cell's nucleus here. Then if they can develop into embryo, this embryo may have exactly the same genetic information of mine. Then this can be practically one possible way of cloning. It seems like this type of nuclear cloning is technically possible, but there is an ethical problem that we need to use egg cells. To avoid using egg cells, maybe cell fusion or in vitro reprogramming can be another alternative approaches. 2012, Nobel Prize was given to this mature cells reprogramming into pluripotent stem cells. The foundation of this study was developed by Dr. Gurdon utilizing frog cells. Later, it was also demonstrated that this kind of a cloning method can be utilized in mammalian cells. And the famous Dolly was born 1996. In this case, the nuclear cloning has been utilized. And here we have 3 mothers. One mother provided egg cells, and then the other mother provide their DNA sources. And then third mother, they take up these fertilized egg cells and then make the baby. Then out of these 3 mothers, which mother is the real mother for this cloned Dolly? Seems like this nuclear cloning is technically possible to be applied to human being too, but efficiency is quite low, and also the ethical problem to utilize the egg cell is a serious problem. That's why the real hero of this Nobel Prize is this person, Professor Shinya Yamanaka. He developed a new method to change any adult cells into pluripotent cells. And this process is called as reprogramming, and this is artificial embryonic stem cell, so-called induced pluripotent stem cells, abbreviated as IPS. Here, let me give you another short movies about IPS.
Most of the cells in our bodies are not stem cells. For example, blood and skin cells have become specialized to perform a specific function in the body by a process known as differentiation that's normally not reversible. However, if some adult cell types are taken, grown in plastic dishes, and given specific genetic instructions, over time, a small number of these cells will reverse from their differentiated state and develop the ability to re-differentiate into any cell type in the body. This newfound ability is known as pluripotency, and therefore these cells are known as induced pluripotent stem cells, or IPS. The creation of IPS cells from individual patients may one day allow us to develop replacement tissues that are a perfect genetic match to the patient. In addition, the creation of IPS cells from patients with specific diseases will allow scientists to develop new drugs to treat a wide variety of disorders.
It's cool, isn't it? Now let's look at the historic paper of IPS development. If this is your project, how would you approach? How can you design to make any adult cells into embryonic stem cells? This is similar to making old cells into younger cells. Sounds like it's a really crazy idea, and their approach is also kind of a crazy approach. In the beginning, they compared the gene expression of different cell types, utilizing embryonic stem cell as a positive control, and compared their mRNA expression level with different type of differentiated tissue cells, and then collected some genes which are only selectively expressed in embryonic stem cells, and assumed these highly expressed genes in embryonic stem cell is important factors to make any cells into stem cells. Sounds like a really crazy idea, but when they tried to put 24 different genes at the same time, seems like there's some observation they found. They make stem cell-like properties. And then they try to take out one by one and try to figure out which is most important factors. Eventually, they found 4 factors. If you express these 4 factors inside the cell, then they can make adult cells into embryonic stem cells. And these 4 critical factors they call Yamanaka factors. Utilizing these Yamanaka factors, and if they grow fibroblast and then transfer these genes into this cell utilizing viral factors, and then this gene can be incorporated into their genome, and then slowly they change their fate into stem cell-like cells, so-called IPS. This result has been reproduced by many different labs and seems like a very robust method. Some left problem is they utilize the viral vectors. That means this viral vector can change their genome structure. This is genetically modified cells. And also one of those factors are oncogenes. That means maybe this CMYC can increase the chances or possibility to make these cells into tumorous cells. That's why whether this artificial IPS can be utilized for therapeutic applications, we'll see. The original Yamanaka method, they utilized retrovirus, or later some other lentivirus has been used. These viral vectors, they have a high efficiency for the gene transfection. The problem is they incorporate these genes into the genome. That's why genomic structure is changed. That's why later another virus which does not change genomic structure, adenovirus, has been used. Even further, utilizing plasmid, temporal transfection also has been tried. Seems like a successful, but general efficiency was lower than retrovirus. The next trial was, rather than genetic incorporation, how about utilizing this protein itself? So make those 4 factors of protein and then transfer that protein into the cell and change the cell fate into the IPS. Also technically seems like possible. Still, easily you can imagine that transferring those huge size of protein into the cell, it may not be so easy. Of course, the best way is utilizing small molecule, something like this. So in the beginning, to replace one of those Yamanaka factors with a small molecule has been tested and seems like it's quite successful. And many of those small molecules, interestingly, they are epigenetic regulators. If you see this table, you can easily see DNA methyltransferase inhibitor and HDAC inhibitor or HMT inhibitors. They change the DNA methylation pattern or histone modification patterns. If you think about the development pathways, it makes sense. Embryonic stem cell mimics the blastocyst stage of an inner cell mass, and those stages, they had a very low methylation pattern, maybe because they needed to express many genes. And if these epigenetic regulators help to activate those gene expressions, the cells can become stem cell-like status. While these small molecule epigenetic regulators are proved to be helpful, still this is kind of complementary to genetic informations. Maybe the eventual goal is only utilizing small molecules and change cell fate from normal cells into stem cells. Finally, the first report has been made utilizing only small molecules. Now it seems like it's possible to make IPS. And these are the 4 molecules which are claimed to make normal cells into stem cells. As they utilize the only chemicals, this IPS they call chemically induced IPS. When they added 4 these molecules into fibroblast, they change its fate, day 12, day 20, day 32. This CIPS becomes more and more close to natural embryonic stem cells. The next year, more exciting paper has been published in Nature. Even without using many chemicals, just treating the cells with slightly acidic conditions, then the cells can change its fate into stem cells. And this method has been called as a STEP. If this is true, then making stem cells are super easy. It may have a high potential to be utilized in daily clinical treatment. Unfortunately, the procedure could not be reproduced by other people, and it seems like there's some procedure problem of this paper and eventually retracted from the journal. Even though there was some mistake here and there, stem cell development is near to clinical applications. Now let's see some current situation.
In the past 50 years, stem cell research has led to some groundbreaking therapies, including bone marrow transplantation and skin grafting. Today, stem cell research continues to hold the promise of new treatments for many diseases. However, most of these potential treatments are still experimental. Any experimental therapy must first be carefully tested to make sure it is both safe and effective before it is approved for patients. This testing process normally takes several years, during which time many experimental treatments may fail because they are unsafe, ineffective, or both. Despite this, clinics throughout the world offer untested and potentially dangerous stem cell-based treatments, usually at a high cost to the patient. As a result, these locations have become a destination for stem cell tourism, where hopeful patients travel internationally to seek unapproved stem cell therapies. Before patients spend time and money, they should familiarize themselves with the facts and think twice about obtaining expensive treatments that have not yet been proven safe or effective.
As you have seen, while stem cells provide a lot of promise, especially for very difficult disease or regeneration of totally lost some part of our organs, still there is some technical problem we need to overcome. Another important direction of this stem cell research is understanding cancer stem cells.
Cancer is the result of uncontrolled cell growth. Therefore, most cancer drugs are designed to kill rapidly dividing cells. While these drugs may have saved many lives, the return of disease following chemotherapy remains a common problem in the treatment of cancer. Scientists now know that not all cancer cells are created equal. Rare cells, known as cancer stem cells, do not divide rapidly and therefore are not effectively killed by chemotherapy. These cells have the ability to renew themselves, as well as the ability to produce rapidly dividing cells. Therefore, following a series of seemingly successful cancer treatments, these rare cancer stem cells can linger and are sufficient to reinitiate the disease. By studying cancer stem cells, scientists may be able to design therapies that specifically target them in order to reduce recurrence and improve our ability to treat cancer.
Cancer is a long-standing problem for human beings, and we have developed lots of treatment methods for cancers. Still, many cancers is very difficult to treat, especially after chemotherapy. The recurring cancers is very difficult to handle. Hopefully, using this cancer stem cell concept, we can come up with new approach, new concept, new treatment to clearly remove old cancers from our body.